our notes for this unit. That is Chapter 13, Genetic Technology. You don't need to know all of this, so we'll just highlight the parts that are really important that you do need to know. First thing is recombinant DNA technology. Remember, recombinant DNA technology is actually one of our vocabulary words, and it is the same as genetic engineering. One thing that we can do with recombinant DNA technology is selective breeding. What this does, it increases the frequency of certain genes into population. This can take many generations, but what it does is it can allow certain organisms to have certain traits that we find desirable. So recombinant DNA technology, again, is what we call also genetic engineering, and it's by connecting or recombining, that's where they get the word, fragments of DNA from different sources. The organisms that are created are called transgenic organisms. They're plants and animals that contain functional DNA, recombinant DNA from an organism of a different type. Alba is an example of this. This is a rabbit who actually was injected with the fluorescent protein found in a jellyfish, and now it glows when in the correct lighting. To make a transgenic organism, and you kind of need to know this, and it goes over a lab that we are doing. You have restriction enzymes first cut the DNA. That's, again, one of our vocabulary terms. Restriction enzymes are usually bacterial proteins that cut the DNA strands at places along the strand in between different sequences. If you did the lab where we actually did the DNA profile, then you would have been able to see this. Some restriction enzymes will cut straight down, others will unzip it, others will cut it in a kind of a zigzag edge, and what it does when we do it that way is it leaves what we call sticky ends, and those sticky ends attract to each other. So if I have two pieces from two different organisms, those sticky ends are going to attract to each other, and they're going to, one will then be inserted into another. This is a good picture and example of what you're doing there. So if you notice that it kind of cuts it so that you have ends that are open, and the sticky ends are going to come together and be attracted. So now I have DNA from two different organisms, and they are now as one. Vectors is what we use to transfer the DNR, and it's a vector means that the DNA from another cell is carried into the host cell. And we usually use biological vectors, and we usually use viruses or plasmids, or we can also use mechanical vectors where we physically insert that with micropipettes or microscopic bullets. Third, both the host and cell's DNA and the foreign DNA have to be cut with the same restriction enzyme, so the sticky ends will match up. The foreign DNA then becomes part of the host cell's DNA. We can use this to carry the DNA into something else. If we want to insert new DNA into a body, etc., we can put it into one of these cells, host cells, and then put it into the body. This is a great picture of how we can combine these different things and then how they get passed down from generation to generation. It can be very, very important, especially when it comes to crops and livestock, that type of thing, in improving our food supply. We can make plants more resistable to certain climates, to be more productive in terms of the vitamins, minerals, and things like that it has in it. It has very, very promising. What else is technology used for? It can be used in forensics, using for crime scenes to diagnose, or also to diagnose infectious diseases. DNA frequency is sections of DNA that we use to find a specific pattern, and these patterns are unique into individuals. A genome, all of the genes of an organism, and that is one of our vocabulary terms. The human genome is really important because we can get exactly where genes for certain traits are on chromosomes, and then we can then use that information later. Linkage, you don't need to know this, but it's just really basically a map of all the genes on the chromosome. Diagnosis of genetic orders is important because if we know somebody has a disorder and then we compare their genes with somebody who does not have the disorder, we can see if it's actually a gene disorder, if it's genes that are causing that. Once we know, is it possible for us to use what we call gene therapy to be able to correct that in a person? So a person might have a genetic disorder, and so can we insert normal genes into their DNA to correct that disorder? It's still very experimental, but it is very, very exciting stuff. little cartoon here for gene therapy. DNA fingerprinting is used in crime scenes and maternity testing, that type of thing, because it's unique. So it can be used in that, and that's what we hear a lot about. It's something that we can kind of understand. All right, that's all of those notes for today. I knew that would be a short one. It would be a good, easy way for you to do your fill notes. This is the stuff that you're also going to need for the lab today and also for your tests coming up.